Well, we are about to hear our last team presentation. I'm asked to give you a little bit of bookkeeping or housekeeping, and that is that after this presentation, we will reconvene for the individual presentations at 12:30. So we will be back here at 12:30. As I said, we're here to hear the last of the team presentations. This is in the area of biochemistry. David Liu is from the Mills Godwin High School. Alan and Jason Lee are from Millburn High School, all in New Jersey. Their mentor is Dr. Jason Chen, professor of biochemistry and molecular biology at VCU, Virginia Commonwealth University. Gentlemen?
Hi, my name is David Liu, and these are my partners Alan Lee and Jason Lee, and our project was the rationale-based design of a targeted therapy for prostate cancer with SPOP mutations. So what do these three men have in common here? We have here current Secretary of State John Kerry, baseball legend and previous Yankee head coach Joe Torre, and movie star Robert De Niro. Well, all of them in the past have been diagnosed with prostate cancer. Actually, American men have the highest number of incidences worldwide, as shown by this graph. In advanced stages, prostate cancer commonly spreads to the bone, becoming incurable. If you look at the PET scan in the middle, the bright shiny yellow dot is the prostate cancer. Since this patient has metastatic prostate cancer, it has spread to the bone, becoming incurable. Therefore, we need to have a better understanding of the mechanisms of prostate cancer in order to develop a new therapy. So what do we know about prostate cancer so far? Well, there has been a lot of progress in the next generation sequencing of prostate cancer. In 2011, Nature published the first whole genomic sequencing of prostate cancer. In it, they found multiple chromosomal rearrangements and gene deletions. And the next year, Nature Genetics published the exome sequencing for prostate cancer. They found that SPOP mutation was the most common mutation in prostate cancer in about 6 to 15% of all prostate cancer patients. SPOP is a speckled type POZ protein and a COL3 ubiquitin ligase. This means that it adds a ubiquitin chain to proteins that need to be degraded. The proteasome is able to recognize this chain and degrade the protein. Interestingly, all SPOP mutations were located in the MAF domain, also known as the substrate binding domain. If you look at figure B, the highlighted red is a mutated residue. We believe these mutations impair substrate binding and can lead to the loss of function and therefore accumulation of substrates. Therefore, we need to learn what substrates of SPOP there are. We conducted a PubMed search and we found 3 of them. The first one was GLE, a transcription factor of the Hedgehog pathway that interacts with androgen receptor and regulates its transcriptional activity. Next, we found SRC3, a co-activator of the transcriptional complex of androgen receptor. Lastly, we found DAX, a cell death regulating protein. We also did an NCBI database search through structural modeling to find 3 peptides that bound with SPOP, and their sequences are listed right here. We identified a consensus sequence between 3 of them, which is, in this case, acidic residue, such as aspartic acid, followed by a string of serine and threonine, and we conducted a blast search with this sequence. After conducting this blast search, it came back with a lot of proteins that had this sequence. So we had to go back and do some more research to find out which proteins interacted with SPOP, androgen receptor, and prostate cancer. What we found was AWP1, a zinc finger protein. So how does AWP1 relate with prostate cancer? We found that AWP1 interacts with PRK1. It stimulates PRK1, resulting in the ligand-dependent superactivation of androgen receptor. PRK1 also promotes the formation of a complex comprised of androgen receptor and other co-activators. Now I'll pass it on to my partner Jason, who'll talk more about a working model for SPOP.
So we propose a model to explain how SPOP mutations could lead to the facilitation of prostate cancer. In this diagram, SPOP has no mutation and maintains its normal activity. Normally, SPOP can induce the ubiquitination of its substrates, such as GLE, AWP1, and SRC3. SPOP adds a ubiquitin tag to these proteins and degrades them through the proteasome. Thus, the androgen receptor complex is not superactivated under normal conditions. However, when SPOP faces a loss-of-function mutation, it is unable to degrade these proteins because they do not fit in the substrate binding pocket. Because these proteins are not degraded, they accumulate in the androgen receptor complex and enhance the androgen receptor activity. We believe that this is one of the mechanisms of prostate cancer. So based on this model, we wanted to develop a new targeted therapy for the inhibition of this pathway. This could be a potentially new type of treatment because current hormonal therapies are based on androgen deprivation, and in most cases, these patients become resistant to this type of hormone therapy. So we thought of 3 potential approaches for the inhibition of this pathway. The first approach is to inhibit the interaction between SRC3 and the androgen receptor. The second approach is to block the interaction between AWP1, PRK1, and the androgen receptor. However, in both of these approaches, it is hard to develop a drug to block this protein-protein interaction. So this led us to think of our third approach, which was the inhibition of the PRK1 kinase activity. So PRK1 is a serine-threonine kinase, which is easier for us to inhibit due to the kinase properties. And after thinking of this approach, we wondered if there was a PRK1 inhibitor already out there. So we searched the database PubMed and found lusartanib, which was a compound identified by Kohler's group using computer virtual screening based on PRK structure. In this diagram, lusartanib docks in the ATP binding pocket of PRK1 and suppresses PRK's activity. Lusartanib has a low IC50 at 8.6 nanomolar and is known to suppress the androgen-induced gene expression. In Kohler's experiment, he tested several different genes, 3 of them being shown over here, that can be activated with the addition of androgen. So in the absence of lusartanib, androgen stimulation significantly induced the androgen response of the genes, as shown by the first black bar over here. However, in the presence of lusartanib, this induction was significantly suppressed, as shown by the next black bar over here. This pattern was similar for the genes that Kohler experimented with and also suggests that lusartanib has the ability to suppress the androgen receptor activity. So we purchased lusartanib and tested it in 3 prostate cancer cell lines to examine its potency with MTT assays. LNCAP, which is a known androgen-sensitive cell, was the most sensitive cell line in our study with an IC50 at 94 nanomolar. PC3 and DU145 were less sensitive to androgen, with PC3 having an IC50 at 920 nanomolar and DU145 having an IC50 at 2300 nanomolar. During incubation, we also noticed that the cells treated with lusartanib developed vacuoles. However, the mechanism of this process and the cell death is unclear at this time. Additionally, we wondered if there's any SPOP mutations present in these 3 cell lines. So to test if there's any mutations, 2 exons covering the substrate binding domain of SPOP were amplified by PCR and sent for sequencing. However, the results from the sequencing did not show any SPOP mutations present in these 3 cell lines. So I'll now pass it on to my partner Alan to talk about lusartanib's effect on the SRC3 protein.
Using Western blotting, we investigated the changes of protein levels in the whole cell lysates after lusartanib treatment in the 3 cell lines. PRK1 did not show any changes after treatment. However, SRC3 demonstrated a significant decrease in LNCAP cells with minimum changes in DU145 and PC3 cell lines. This coincided with prior findings that LNCAP cells were the most sensitive to lusartanib. Nevertheless, we were puzzled by these results because lusartanib is a PRK1 kinase inhibitor, and based off this mechanism, it should not have had any effect on SRC3 protein levels. Therefore, we conducted further examination through the literature and found that an atypical PKC phosphorylates SRC3, which prevents it from being degraded by the proteasome. Lusartanib inhibits the atypical PKC's function, blocks the phosphorylation of SRC3, and thus facilitates the degradation of the protein normally. This possibly explains the puzzling results. We wanted to further examine the effects of lusartanib on protein levels in the different compartments of the cell, so we conducted subcellular fractionation by differential centrifugation. Here are the Western blotting results for the subcellular fractionation. Localization control, histone H3, was used to confirm the separation of the nucleus from the cytosol. In the nucleus, SRC3 did not show any changes after treatment. However, in the cytosol, SRC3 demonstrated a dramatic reduction in LN cap cells with slight decreases in DU145 and PC3 cell lines. This is consistent with the fact that the degradation of SPOP substrates by the proteasome mainly occurs in the cytoplasm. PRK1 actually showed a slight increase after lusartanib treatment in the nucleus. This is possibly due to a feedback loop mechanism, which needs to be further examined. In conclusion, we started from SPOT mutations detected by the exomic sequencing, understanding the biological significance of these mutations, to searching for potential SPOP substrates using a PubMed and BLAST search. Key SPOP substrates that may be associated with the formation of prostate cancer include GLI, AWP1, which interacts with PRK1, and SRC3, all of which are tied to androgen receptor activity. Using this pathway, we rationally designed a targeted approach using lusartanib, a PRK1 kinase inhibitor. The feasibility of using lusartanib is supported by our cell line study. Their sensitivity is associated with the decrease of SRC3 protein after lusartanib treatment in LN cap cells. In the future, the complex comprised of SRC3, androgen receptor, and AWP1 PRK1 will be a useful target for new drug development in prostate cancer. The major application of lusartanib is in hormone-sensitive prostate cancers based on the current data. A future combination study using lusartanib and hormone blockade could be a potential new effective therapy for treating hormone-sensitive prostate cancers. As you can see in this diagram here, this method provides 3 hits to this pathway, as opposed to the current method of only hitting the androgen receptor. We would like to thank our mentor, Dr. Jason Chen, for all his guidance and providing us with a lab to work in. We would like to thank Dave Durant and Dr. Ray Lee for all their technical instruction and clinical input in prostate cancer. We would like to thank our families for all their support throughout this project. Most of all, we would like to thank the Siemens Foundation, College Board, and George Washington University for hosting this competition and having us here. Lastly, this experience has been amazing for all of us here. And there are a lot of things that we can all say about this, but we feel it is best represented by this quote from Wayne Gretzky, one of the greatest hockey players of all time. "You miss 100% of the shots you don't take." Thank you.